In this mini lecture, we're going to talk about the Lac Operon and understand it a little bit. E. coli, which are prokaryotic cells, they encounter many different types of sugar in the environment, and some of which could be lactose, some of which could be glucose. And whether it's lactose or glucose, they need to express genes, proteins, in order to actually break down those sugar. So a Lac Operon or an operon is actually a unit that actually functions together in order to regulate or break down lactose, which is one of the sugars that E. coli tends to break down. So in a nutshell, what you need to understand first is the component of this Lac Operon. You have a promoter, you have an operator, you have a repressor, which is also sometimes referred to as the Lac I. Then you have the genes, which is, or in this case, we have a Lac A, Lac Z, and a Lac Y. So from the picture that I have somewhere here, which you're going to see, we also have the addition of a CBS region where there is actually another molecule known as a trans factor cap that gets to bind in. But the reason why we have this trans factor cap as well is that because glucose is the preferred sugar for E. coli. So what they want to do, although they have to regulate or they have to break down the lactose, but they give preference to glucose first. So what do you see? A promoter, we already know the role of a promoter in transcription. That is a very important region, a promoter region, where polymerase eventually comes to bind and result in transcription. But an operator in this scenario here actually work hand in hand with a repressor. So how does this work? If you look at the picture below, is that in the presence of lactose, you actually have a lactose that is going to bind to this repressor. And as a result, the repressor is not going to be able to sit in your operator. So this is like a switch button on and off. If this is your, let me just put it here. So if this is your operator, when you have lactose, your repressor will not be able to bind. But as soon as you don't have lactose anymore, your repressor binds like this to your operator, and as a result preventing the transcription and eventually the production of the Lac A, Lac Z, and the Lac Y. So the cap trans factor, or the CMP, the way it works is that now this is also interested into the regulation by actually causing further gene expression of those Lac A, Y, and Z. But that is only in one condition, in condition where the glucose level are really low or not present. As you're going to see from this picture that I have now, in the presence of glucose, there is no activity, so ATP and cyclic AMP is not going to interact. As a result, the cap is not going to bind in this allosteric site near the promoter and the full operational unit. As a result, you are still going to get the Lac A, Z, and Y, but it's not going to be in abundance. But as soon as the glucose level gets depleted, what we see is that now there is an interaction between the ATP and the cyclic AMP, and as a result, the cap is going to bind to the CBS site and this allosteric site, and as you see from the number of product that we're producing on the other side, we actually have enhanced our production of lactose. So that is in a nutshell our understanding of the Lac Operon and how it regulates the expression of genes that are involved in the breakdown of lactose.